Hey guys, last video, last set of notes for cellular respiration. So let's get this shared and presented. So we really have focused on how does cellular respiration run in the presence of oxygen. But what happens if there's not oxygen present, or if you don't have a mitochondria, or you get the concept there? So we're going to look at respiration without oxygen. Remember, in cellular respiration, oxygen is the final electron acceptor. It drives the electrons down the electron transport chain. So how do organisms produce ATP in the absence of oxygen? The answer is anaerobic respiration, fermentation. They're kind of the same thing, just the directions, the pathways are going to vary. So anaerobic respiration generates ATP using an electron transport chain in the absence of oxygen. This specifically takes place in prokaryotic organisms that live in environments with no oxygen. So no oxygen, no mitochondria. The final electron acceptor in these specific organisms is either going to be sulfates or nitrates. Now, like I said, anaerobic respiration and fermentation are very similar. Fermentation generates ATP without an electron transport chain. So it basically is an extension of the glycolysis process, because for whatever reason, these organisms cannot move into the mitochondria or do not have a mitochondria to continue with that reaction. In this case, we're going to recycle the NAD+, and it's going to occur in the cytosol just like glycolysis does. And remember, we said this is a situation where no oxygen is present. There are two key types of fermentation that we focus on. One is alcoholic fermentation. The other is lactic acid fermentation. Okay, so we're going to start with alcoholic fermentation. So we had our glucose, we did glycolysis, and we have our pyruvate now. Because oxygen is not present, not available, we're going to go through a further set of chemical reaction pathways that will release 2 carbon dioxides and form a substance called 2-acetylaldehyde. This will then use the NADH that we had from glycolysis. It's going to utilize that to break the acetylaldehyde into 2 ethanol molecules. Yes, alcohol. Alcohol fermentation, the pyruvate is converted into ethanol. Bacteria and yeast in particular will do alcoholic fermentation. The other method was our lactic acid fermentation, and this actually happens within us. So in lactic acid fermentation, pyruvate is reduced directly by NADH, just like the other, but this one forms something called lactate. So our 2-pyruvate get reduced by NADH and they form lactate or lactic acid. Our muscle cells are a key example of where this happens in the human body. So when you're working out, it tends to be a situation where the muscles are going to run out of oxygen because you're using the oxygen faster than it can be replaced in the body. So they can switch over and use lactic acid fermentation to produce ATP. This is what causes that burning sensation that you often feel when you're performing strenuous exercises. And then, of course, eventually that pain goes away after time because you would then break down the lactate. So the muscles produce the lactate, it goes into the blood and then gets broken down back to glucose in the liver. Part of the concern is that when lactate is in the blood, it lowers the pH, and your blood's pH has to stay within a certain range, or you could become extremely ill and possibly die. So you want to be careful not to overdo it and build up too much lactate so that it lowers your pH too much. So if it builds up and it cannot be broken down properly, we end up with what we call lactic acidosis. It's going to make the pH of our blood too acidic and ultimately can make you very sick. Make you very sick or lead to death. So let's kind of put the whole picture together. So glycolysis happens in the cytosol, all living things. If there is oxygen present and mitochondria, we go through pyruvic oxidation, citric acid cycle, and oxidative phosphorylation, and we can make somewhere around 30 to 32 ATPs. When there's no oxygen, we have fermentation, which could either be lactic acid fermentation or alcoholic fermentation, and each of those will make 2 ATPs. Obviously, the process with oxygen is much more effective in providing us with energy from the glucose. Okay, practice free response. The catch and release method of fishing was once thought to be a beneficial method for conserving the number of fish in a body of water. But it is now known to be potentially deadly for up to 72 hours after a fish has been caught. When a fish is caught and pulled out of the water, it cannot take in oxygen. The fish experiences extreme stress during this time, which triggers shimmying, a condition where the fish will use their muscles to vigorously shake back and forth. So I want you to identify the stage of cellular respiration that is directly affected by the lack of oxygen. Explain the importance of oxygen for cellular respiration. And then explain how the fish is most likely able to generate energy to shimmy in the absence of oxygen. So pause and write your answer. Okay, and here's our next practice free response question. Examining the permeability of mitochondria revealed that the outer membrane was permeable to hydrogen ions. After discovering this, a researcher wanted to study the effects of pH on the activity of mitochondria. Mitochondria for liver cells were isolated and exposed to solutions at pH 9, 8, 7, 6, and 5. The results indicated that mitochondria were most active at pH 5. Explain how pH may affect the relative concentration of ions in the mitochondria, and propose a model to explain how a low pH could increase the productivity of mitochondria. Hopefully that kind of gives you a better idea of what's going on with cellular respiration. Bye.